It's a great pleasure to be here, and this is a beautiful room, and it's very impressive to know all the very important people who have spoken here before. And what I wanted to do today is to give you all a sense of the problems that are mostly preoccupying my scientific brain these days. And some of you have seen a lot of these data, but I'm sure that there are many of you who've seen it and forgotten it, and other people who are the younger people in the audience who may not have seen it. But I think the nicest part about giving a longer talk is that one can actually give people a sense of why one's thinking about what one's thinking about and how you got there. Anyway, just before we leave this slide, this is one of the images from one of the little e-life opinion pieces I write. And so this is a very beautiful image that my nephew, who's an artist, made. And he illustrates all my pieces, which is partly why I write them, because they pay him. And you know, if you're a starving artist, it's good to get paid. Okay, so before getting going, I would just like to say I've been extraordinarily privileged throughout my scientific career to have truly fabulous people working with me. And I think I can go back to many of the really important milestones of the work we've done, and I know exactly where the genius and creativity of the people who worked in my lab or with us, how they contributed to those pieces. And I think that one of the most, and this is just for me talking to all of you, one of the most precious parts about being a PI is that you know that the work you're doing is much, much bigger than it would have ever been if you were doing it by yourself. And I really value enormously all of those people. And I'll try and mention their names as I go. So the problems that I'm really preoccupied with today are basically these two problems, and they date back a while in our work. One of them is understanding individual variability in circuit parameters and circuit function, and the second is the role of homeostatic control systems for maintaining stable brain function and resisting perturbation. And both of these are fundamental problems that are as important for the human nervous system as they are for the nervous system of a crab. And they deal with issues that immediately pop into your life if you spend many, many, many years recording from the same identified neurons in a quote-unquote simple preparation, which isn't so simple. And you discover after 25 or 30 years of recording from the same LP neuron that it's not always the same. And then that starts, and eventually you start taking those data seriously. Likewise, the second problem is one that dates back to about 1992 when we were working on trying to do biophysical analyses of single neuron dynamics. And we were starting to build models, and the models we were building were extremely fragile. And they depended very heavily on all the things we could measure the least well. And this made me extremely upset. And I realized that for us to tune this model, we had to get exactly the right relationships of current densities between all the different ion channels in the cell. And I realized that we had no understanding about how that came about. And after building a model that took 9 months of a really smart postdoc and graduate student working along to get a very not great model, which was as good as anyone in the field at the time, but it still was not great, I looked at Larry Abbott and I said, how come the LP cell can do it every time and we can't do it at all? And he said, that's because it's a really hard problem. And I said, well then, what's the answer? So anyway, this is a problem. So when we come back at the end of my talk to this whole issue about how cells control their intrinsic excitability over very long time periods, even though the lifetime of channels and receptors is turning over very quickly. This lets me to start thinking about the fact that not only do nervous systems have to build themselves during development, but they have to be constantly rebuilding themselves throughout the lifetime of the animal. And so the yin and yang of trying to allow mechanisms for development, modulation, flexibility, experience-dependent changes, but how that had to be superimposed with all the mechanisms that maintain stable function and how you have those interact is something that we've been thinking about for quite a long time. So these are related problems all about how you have mechanisms such as neuromodulation, et cetera, to produce flexibility, but how you interface those with maintaining healthy, stable function. Okay, so the system that we work on is the crustacean stomatogastric nervous system. And this nervous system comes from lobsters and crabs. What we do is we buy the animals from whatever local fishermen we can get a hold of, and who, of course, refuse to tell us where they got the animals from. But that's another whole drama. And then I used to, they do now, people in my lab, we take the stomach out of the lobster or the crab, and lobsters and crabs have a very complicated stomach. There are teeth inside of it. There's a very complicated filter. There are 42 sets of striated muscles that are moved by a series of motor neurons in the stomatogastric ganglion. And so it's a very complicated mechanical device, which is under neurogenic control. And we dissect the stomatogastric nervous system. That includes the single stomatogastric ganglion and these anterior commissural and esophageal ganglia that send modulatory inputs down a single input nerve into the neuropil of the stomatogastric nervous system. We desheath the STG, or the stomatogastric ganglion, and record intracellular with sharp electrodes from 1, 2, 3, 4, under duress 5 or 6 neurons simultaneously. We place extracellular electrodes on the motor nerves that contain the motor axons on their way to the muscles which have been removed, which would be several floors down if they were still here. And when we do that, we record here, there are 2 major rhythms produced by this nervous system. One of them is the pyloric rhythm, and this rhythm is seen here in the discharge of LPPYPD, where this is a constrictor motor neuron, this is a second constrictor motor neuron, and this is a dilator motor neuron. This is the valve. And so you have constriction 1, constriction 2, dilator, constrictor 1, constrictor 2, dilator, and the canonical period of this rhythm is about 1 second. And the same neurons can be recorded on that extracellular electrode here with LPPYPD, LPPYPD. Now, in the early 1970s, when Don Maynard basically pioneered this preparation, he was interested in answering 2 questions. Number 1, why is there a rhythm at all? And 2, what determines the actual phase relationships of the relative timing of all the units? And to do that, he reasoned it was really important to actually record the intrinsic properties of each cell by itself, and then work out the synaptic connectivity. So a number of us over the years, by about 1980, we had a connectivity diagram, as it was then called. Today we call these connectomes. Then it was a connectivity diagram. You see nothing much changes. The connectivity diagram was just as disappointing in 1980 as some of the connectomes will be in 2016, but we'll come back to that in a moment. Well, there's a lesson here, right? So on all my diagrams, an electrical synapse is shown with a resistor symbol, and all of these synapses are chemical inhibitory synapses. So the first thing you see is that there are no rapid excitatory synapses. Instead, it turns out that the ABPD, there's 1 AB and 2 PD cells in each ganglion. This 3-cell network serves as a pacemaker. It's pretty close to an intrinsic oscillator. And when these pacemaker neurons depolarize, they rhythmically inhibit the LP and the PY cells. And then because of the intrinsic properties of and the synaptic strengths, the LP cell pops up first. It turns off the PY cell. The PY cell eventually rebounds from inhibition. It shuts down the LP cell, and then the pacemaker starts again. So that was the 1980 word model of how this rhythm works, and it's approximately right. So you might ask, what have we been doing from 1980 to today? I know some of you were thinking that. I would in your shoes, right? So what we've been doing is I'd like to point out that if I showed you this connectivity diagram of this connectome and just showed you this, there'd be absolutely no way you would know what these dynamics are, right? And that's because there's way too much information missing here. What's missing are all the voltage-dependent all the voltage-independent currents in each one of these cells, all the history dependence of the activation that come from the activation of those currents. What's missing is the strength and time course of all these synaptic inputs. And then you would need, even if you had done all the right voltage clamp measurements and measured everything 1 by 1, you would still need some way of putting it all back together again. And that's why at the end of the 80s, I was sort of susceptible to being seduced by computational neuroscientist, because already, even as a graduate student, I had understood that I was very bad at thinking about more than two nonlinear processes at the same time. And what computational models allow you to do is actually put together a lot of nonlinear processes and actually understand how the behavior of the system depends on the interaction of those processes. So what we've been doing, among other things, is trying to really understand how to go from cellular mechanisms of biophysics back to dynamics and then back to changing dynamics. Because obviously all these synapses are modulated and they all show history-dependent changes in efficacy, likewise many of the intrinsic properties. So that just is sort of shaping where we are. But I'm going to show you the second motor pattern, which is the gastric mill rhythm. Now these are all extracellular electrodes, and this is on a much longer time base. This period is about 10 seconds. So the pyloric rhythm, one you just saw, is seen here, LPPYPD on this compressed time base. But you also see LG is depolarizing, firing a burst of action potentials, hyperpolarizing. And along with that, you can see that's alternating with bursts in DG and bursts in GM. And additionally, and this is why I'm showing you this, these two neurons, IC and VD, are firing in phase. When they're active, they're firing in phase with the pyloric rhythm. And they were originally classified as pyloric neurons. But you can also see they're firing in an envelope of activity in time with the gastric mill rhythm. So they are, if you want, multiplexing both rhythms, or at least participate, and they do participate in the generation of both rhythms. So, and we spent a lot of time trying to understand how these neurons and other neurons switch back and forth between part of both rhythms. But the reason I'm showing you this is because this is actually the connectome of the full Crab stomatogastric nervous system. And if you look at it, you see that there's no clear demarcation between the neurons that are part of the pyloric rhythm and part of the gastric mill rhythm. And if you think about it, there's no real demarcation in the brain that actually says, oh, these neurons are part of this functional circuit, and then there's a little red line, and then there are neurons that are part of a different circuit, and there's a blue line cutting them off from others. At the end of the day, every neuron in the brain is connected to every other neuron in the brain by a path which is probably not that circuitous. But the reason I'm really going to show you this right now is to make another point, and this is a point about parallel pathways and multiple interacting circuits. So if you look around here, you see all these electrical synapses. Some of them are very strong. This synapse is strong. Some of them are much weaker. Some of them are non-rectifying. They say they pass current pretty well in both directions. Some of them are highly rectifying. So regardless, these electrical synapses create a ton of parallel pathways. And I'm going to show you one right here. So if you look at the IC neuron, that's one of those neurons that participates in both rhythms. It receives a monosynaptic IPSP from PD. And then PD inhibits LP, and then through the electrical junction, it can be inhibited polysynaptically. Likewise, interneuron 1 directly inhibits IC and also indirectly inhibits it through the LG in this electrical synapse. Now, Gabriel Gutierrez, who at the time was a graduate student in my lab, noticed that this 5-cell circuit has a beautiful symmetry to it. And she started her life as a physicist, and you know, physicists think about things like symmetry. And so this appealed to her as a modeling project. She originally said she was going to build a model of the whole STG, and that took her 2 days before she decided that was too hard. But then, you know, it's an intelligence test. How long does it take between the time you say, I'm going to build a model of the whole STG, before you say it's too hard? So she's the fastest. I've had other people take a week, other people take 6 months, you know, I mean, anyway. So, but she did notice this 5-cell circuit with this symmetry, and so she said she wanted to build a model that had a hub neuron where she could study the effect of a fast oscillator. Here, this half-center reciprocal inhibition is part of the fast circuit, and a slow circuit, and she wanted to know how these two fast and slow oscillators would fight for the soul of the hub neuron. And so she built a very simple model, and I'm going to use that model to make a few points about parallel pathways. Okay, so she decided to model each neuron with a very simple model. These are Morris-Lacar oscillators. They have an inward calcium current, an outward potassium current. They also have an IH, that's a hyperpolarization, activated inward current. And then they have chemical inhibitory synapses in the network and the electrical synapses. But each cell, now because she wasn't really trying to model the STG, but this was now, the STG was a motivation for this 5-cell circuit, she renamed all the neurons so you wouldn't get confused. And she's got F1 and F2, those are fast neurons. HN is a hub neuron. S1 and S2, these are the slow neurons. And so in isolation, F1 is fast, F2 is fast. They're the same. The hub neuron is an intermediate frequency. S2 and S1 are slow. And when you reciprocally inhibit F1 and F2, they fire in alternation, as you would expect, at a fast frequency. S1 and S2 fire in alternation more slowly. And if you just hook up the electrical synapses with a high enough coupling coefficient, you can see the 3 cells synchronize. And so none of that is surprising. That's all pretty well understood. So now we've talked about what happens when you put the 5 cells together. And here's the 5-cell circuit. And I'm just going to introduce one more piece of terminology in one picture. So Gabriel named the strength of these electrical synapses GL, conductance of the electrical synapses. These are called G syn B. She's calling these synapses synapse B. These are synapse A. So she's going to first look at what happens when you change the effects of G syn A and GL on the dynamics of the network. But she is a very clever young woman, and she wanted a way to visualize the frequency of all elements in the circuit at the same time. And so she came up with this really nice way of graphing it. So this is 5 concentric circles and squares, where the 2 outer circles are the frequency of the F1 and the F2. The square is the frequency of the hub neuron, and the 2 inner circles are the frequency of the slow. So over here, with one set of parameters, you can see the 3 inner boxes and circles are slow, they're blue. This is on a heat map, and they're firing slowly. And F1 and F2 are firing more rapidly. And then in the different set of parameters, all 5 cells are firing in the same rhythm, and it's an intermediate frequency rhythm, so they're all the same color and they're all green. Okay, so now we're going to use this way of visualizing things to see a full parameterscape. What Gabrielle is doing is she's plotting this G syn A on the X and G electrical on the Y. And you can see these enormous, and now we're using that notation, which allows you to see different states of the network. So over here, you've got 1 fast neuron and 4 slow neurons. Over here, you've got 5 neurons that are all firing in the same intermediate frequency. Over here, you've got 4 fast neurons and 1 slow neuron. And you can see how the system changes its behavior as a function of these 2 synapses. Now, I'm not going to spend a lot of time talking to you about the details of why these state changes or bifurcations take place, although we understand them pretty well. Instead, I want to use them to make a very simple point. So imagine now that you're studying the effect of a modulator, modulator A. And modulator A influences the strength of this synapse. So if you start with a network over here, and you modestly increase the strength of this synapse, you actually will go through 2 different state changes. So the circuit will completely change its qualitative behavior as a function of a small change in G syn A. However, if you had started over here and now had a much bigger effect of modulator A, you can see that much larger effect of the modulator produces no qualitative change in the system's behavior. So this is a very discrete example of what some of us have been calling state-dependent neuromodulation, which just articulates in a very easy to understand way why it is that a big change in synaptic strength might sometimes produce very little change in behavior or circuit output, whereas other times a very small change in a synaptic strength produced by a modulator might be completely transformative. And obviously, you could go in the other direction. But that's not really why I'm showing you this. This is all set up. The next slide. So what Gabrielle did here is she noticed something. And this goes back to parallel pathways. This is a 5-cell circuit. And she noticed that if she started out with one set of parameters here in this control, A, you have 2 fast and 3 slow cells. And then she decreased the strength of G syn A, and she flipped HN from slow to fast. Then she went back to those parameters, and she decreased the strength of this electrical synapse, and she again flipped HN from slow to fast. And then she did an entirely different manipulation, and she decreased the strength of these synapses, and again she flipped HN from slow to fast. So the take-home lesson of this is you've got a 5-cell circuit, but it has parallel pathways. That is to say, there are multiple ways that cells can interact. Because of those parallel pathways, the circuit is degenerate. That is to say, there are multiple mechanisms by which you can go from state 1 to state 2. Now, sometimes when I talk about this, I make a joke and I say, there's a really smart graduate student here at the Karolinska who's doing this experiment and gets this result and goes to his advisor and says, look, I found the mechanism that accounts for this change in behavior. And they publish a paper saying, the mechanism for X. And then the graduate student at UCSF, who's doing this experiment, says, those people at the Karolinska are just stupid. Don't they know I've found the mechanism, right? And then both of them published their papers in Nature because, you know, they were first. And some poor graduate student in Singapore or Mumbai does this experiment and says, but I have it, and they're all wrong. And actually, they're all right. And they're all right, but they're all only seeing a piece of the principle. And so every time you do an optogenetic or a manipulation, a pharmacological or an optogenetic manipulation, you always have to bear in mind that if there are parallel pathways in your network, there almost certainly are multiple ways that those transitions can take place and do take place because the brain is too smart to depend on a single mechanism to do anything important, right? And so if you remember nothing else from what I say, remember, the brain is too smart to depend on one mechanism to do anything important. And this is probably true in all parts of our brain, that there are multiple ways to do any change in behavior if it really is important to the animal. Okay, so one more slide of Gabrielle's. And there's a funny story about this particular image, because this is the image that's it's actually not in the paper, but it's in the little, advertising thing that the journal uses. And I made Gabrielle make this image for that. And I said, why didn't we put this in the paper? And she said, oh, I don't know, because you made it, make it later. Anyway, what this slide shows, in this study, what Gabrielle was doing is leaving the synapses more or less constant and changing the intrinsic properties of the hub neuron. And what she was doing is she chose the hub neuron, and then she changed the conductance densities, the calcium and potassium currents, so that it changed its waveform, but she found solutions that had the same frequency. So these are all neurons that are going at the same frequency, but they have different waveforms. And then she embedded these neurons. So now we're looking at modulation of intrinsic properties, in this case the waveform. And then she embedded these neurons into the same network. So here she embedded these neurons into this network, and as she changed the waveform of the isolated cell, you can see that the network is basically unmodified. So the network's behavior is more or less insensitive or robust to this change in the waveform of the HN cell. And then she looked at a second set of network parameters which are different. So here this synapse was big, there it's small, here the electrical synapse was small, here it's big. And under these conditions, as she modulated HN, you can see the network completely is transformed. So in this case, modulation of HN only affects HN. In this case, modulation of only HN completely changes the behavior of the entire network and their relationships. So this, again, is a state-dependent modulation. It's a different form of it, but it tells you that depending on the starting set or the starting set of parameters, modulation of a single neuron can ramify throughout the entire network to which it's connected, or it can be restricted to the relatively single cell. And this almost certainly is important as you start thinking about large circuits as well. Okay, so moving back in time, I just want to remind you that the behavior of single neurons, this was sort of embedded in what I've talked to you about, the behavior of single neurons depends on the balance of conductances in the membrane. This is just a picture that Jung Liu, who was a graduate student of Larry Abbott's, made for talks for me. He has a model neuron that has a sodium current, 2 calcium currents, 3 potassium currents, a transient outward current, a calcium activated K, and a delayed rectifier current. And one set of conductances gives you a silent neuron, a different set of conductances gives you a tonic firing neuron, and a different set of neurons gives you a bursting neuron. And that's all very conventional, and there's nothing surprising. What Mark Goldman did next, was to ask how many different ways can you get a burster? How many different kinds of conductances can you find to give you basically the same bursting behavior? And it turns out that, again, this was the first instance in my lab where we discovered that there were multiple sets of membrane conductances that could give rise to basically the same electrophysiological behavior. And this allowed Astrid Prince then to ask that same question at the network level. When Astrid, she wanted to build a good model of the pyloric rhythm. This was one of the earlier ones. And so she spent a week trying to hand-tune individual neurons to capture the dynamics of the pacemaker kernel, the LP neuron, and the PY neuron. And after a week of really bad things coming out of her mouth that were in German, so I don't really know what she said, she went silent. She got very happy. And then I discovered that she had all these models running on every computer in the lab, because what she decided to do was to take her model neuron that had 8 currents, and then to take 6 values of each of the 8 currents, simulate all against all. So she built a database of 1.7 million model neurons, and then she searched within that database for all the models that would be good ABPD cells or good LP cells, etc. So from that, she chose 5 pacemaker neurons, 5 LP neurons, 6 PY neurons, and then she chose either 5 or 6 values of the synapses in the network, and then she again ran all against all. Now this by now, I had thrown her off all the computers in the lab onto the cluster, and so it ran happily in the cluster, and she ran 20 million model networks. And then she asked, how many of these are good pyloric rhythm models, or adequate? So starting with her 20 million, 19% of them were pyloric-like. That means they were triphasic in the right order. And then of that, 2.4% of them were what she would call a good pyloric neuron, a good pyloric network model, which means they were triphasic in the right order, and they fell within the range of the values that Dirk Bucher measured from 99 lobster preparations. So this was an absolute an absolute sort of shift in strategy. It was actually a paradigm shift, because instead of trying to fit a mean set of data or a canonical image or a canonical version of what we thought the system was doing, Astrid said she wanted to find all the models that would fit within the range of the behaviors that the biological system was doing. So we were looking for a family of models that would capture the same distribution as the range of behaviors in the biological system. And so she used burst duration, cycle period, number of spikes per burst, and a variety of behaviors to select the successful models. And I'm going to show you 2 of those successful models, because this paper was actually completely transformative of the work coming out of my lab. And I like to go back to it because it's an example of a theory paper which changes your way of thinking about something so that you end up doing different experiments for 5 or 10 years. And that's the kind of theory paper which I think is really important. Not that it's a good model of pyloric rhythm, it isn't, but that it showed us something that meant we were changing what we were going to do. Okay, so here are two model networks. You can see they're pretty similar. And if you were an electrophysiologist and you had recordings that were this similar, you'd say, yeah, those are two healthy sets of recordings. And these are the underlying conductances for some of the underlying conductances. And you can see this set of conductances and this set of conductances are entirely different. Here, the H current, the PY neuron is big, here it's small. The PYLP synapse here is big, here it's small. The ABLP synapse is big, here it's small, etc. So you can see this set of parameters and this set of parameters are entirely different, and yet they're producing more or less the same motor pattern. So this makes exactly the same point as we made before, that there are multiple ways of producing a similar network output. But this is obviously only a model. And when I started talking about this, biologists actually found it very comforting. And biologists found it very comforting, and I found it very comforting, because it said that the noise, or the animal-to-animal variation, this is where the animal-to-animal variation starts popping up, that we were seeing in our electrophysiological data might be a feature, not necessarily the fact that we were doing something wrong, right? I mean, I never do anything wrong, but that's, if you believe that, I could sell you the Brooklyn Bridge and something else, right? But a lot of theorists were very offended by this. And the theorists at the time who were very offended by it were offended by it for a number of reasons. The bad one was, they said, this can't be true, don't you know that biology is optimal? And I said, what do you mean biology is optimal? I don't know any biological system that's optimal. I mean, you're not optimal, right? I'm not optimal, I mean. So, but they were trained to measure, to calculate optimal solutions, and so they assumed that the biological solution would have to be optimal. And if they were busy trying to fit a single set of traces, they weren't aware of the fact that those traces were selected from other traces that maybe didn't look exactly the same. But they did make one really important point. When we had seen these differences, we assumed that these differences might be because this change was compensated for by something else in the network. And that's probably sometimes true, maybe sometimes not true. Sometimes these differences occur because maybe this synapse doesn't matter how big it is. But what they pointed out, and this is a very important point, is that this set of, if you have this set of parameters and this set of parameters, there will be some perturbation in the universe of all possible perturbations that is going to separate this network from this network. And they kept on saying, and I heard this from people when I was talking, that it doesn't matter that you can find a solution for this static snapshot, but you really have to ask the question, you know, how sensitive are these different solutions to the two perturbations? And I immediately answered, I don't care about the fact that there will be a perturbation in the universe of all perturbations that will separate them. It made me realize that what mattered is, if I took this seriously and thought this was crab 1 and this was crab 2, then I have to ask, could crab 1 with this set of parameters and crab 2 with this set of parameters respond robustly to the environmental changes it's going to see, as in temperature changes or salinity changes or neuromodulation? So a lot of what we've been doing in the last few years has been asking, number one, is how much animal-to-animal variation is there? And then, can animals with variable underlying systems be robust to perturbation? And so just to skip through that, so this led to these questions. How variable are the parameters in real biological networks? And that's why we started measuring mRNA levels and all sorts of things on an animal-to-animal basis. Which parameters are highly variable because they just don't matter? And how reliable are networks of different underlying structures against environmental perturbations and neuromodulation? And this is really the most interesting part of this story. Okay, so question 1, I'm going to skip through very quickly. Dave Schultz and Jean-Marc Gaillard isolated single neurons. First they would measure the network, then Jean-Marc would do voltage clamp in single neurons, measure currents, and then they would dissect the cells out for single cell PCR. This was actually published 10 years ago now. And what you see, This is LP. There's one LP cell in each ganglion. LP1, LP2, those are two different animals. You can see that if you superimpose them, their waveforms are almost identical in the ongoing rhythm. But when Jean-Marc went into voltage clamp, you can see the calcium activated potassium current in this guy was much bigger than it is in this guy. And there was a 2 to 4-fold range in the conductance densities that we measured for every current we've measured. And this holds true for almost every membrane property we've measured since, and this is now many more. And in this first slide, Dave found, again, a 2 to 5-fold range in the mRNA, the copy numbers in identified cells of, in this case, it was 3 potassium channel genes. And the fascinating thing was that the conductance that Jean-Marc measured was highly correlated to the copy number for the channels, for the mRNA, for the channels that give rise to those currents. Okay, so this gave us a sense of what the animal-to-animal variation was going to be, looking at defined parameters in well-identified neurons that are absolutely well-identified. There's only one LP cell in each ganglion. We identified it completely unambiguously. The other thing that came out of this study was that there were very strong correlations between the expression of some of these channels. This is the H current, this was HCN in LP, and it's highly correlated with the values of SHA, SHAB, and SHAL. And here you can see that on a 4-dimensional plot. So the fact that we were doing these data on a cell-by-cell basis allowed us, and that we were measuring multiple things in the same cell, allowed us to see correlations that people were missing because they were pooling data and just comparing means against means. And all of this stuff, the really interesting stuff, would be washed out if you weren't able to keep everything on a cell-by-cell basis. One more of these slides, and I'll just show you. Now, we ended up doing many, many different channel genes in many different cell types. A little bit of it is here. This is the IH to SHAL correlation. It's present in many cell types, but notice the slope of that correlation is different, and that's going to become important at the end of my talk. Okay. So, and I guess I should show you this, and this was just to say that in 2007, when we published this paper, we hypothesized that the set of correlations in channel expression, the black boxes are where the correlations are. You can see they're different in each cell type, and we speculated that those sets of correlations were going to be a part of that cell's identity. And we couldn't prove it at this time, and it's only now that we think we have an explanation for that. Okay, very quickly, talking about variability of a different kind. We've always been struggling with the fact that stomatogastric neurons have very complex morphologies. They're very beautiful. These are 6 GM cells that are actually being shown to you at relatively low resolution that Adrian Topolik, a graduate student in the lab, has filled for other reason. And if you just look at them, you can recognize them as GM cells. They each have multiple axons. They all have a complicated neuropil. They all have a major big process. But obviously, even by eye, you can see the details of where the branches are, et cetera, is different. So Adrian, for a variety of other reasons, set out to use focal glutamate uncaging to look at where on the surface of the cell the glutamate responses are going to be. Now, glutamate is an inhibitory transmitter. It's the major inhibitory transmitter here. So Adrian built a little uncaging microscope that she attached to our dissecting microscope. So she first fills the cell and then uncages at all these different spots, and she can do up to 40 or 50 spots in a neuron. And you can see here at spot 1, purple, wherever purple is, down there, you have a nice inhibition. And then here at spot 5, which is here or here, I can't tell from the colors, you can see there's a big inhibitory response, but spot 4, there's nothing. Spot 6 is a big response. Spot 11, which is here up the cell body, there's nothing. So the glutamate responses are spread out throughout the whole neuropil. And if you then take this image and just do a skeleton reconstruction, you can see the skeleton reconstruction, and then Adrian can actually put those spots back onto the skeleton reconstruction and figure out where the cells are responding. And this allows us to now take what she and Alex Sutton, another student or another postdoc are doing, is use the spots on these skeleton reconstructions to say how much variation is there across 15 of these cells in terms of where the glutamate responses are, which branches, how far are they. And I'm not going to show you all those data, but I'm going to show you something cool, which is, I said to Adrian, at some point, because we all anguish about cable properties, right? That's the biggest nightmare when you work on complicated neurons. And so here's purples down there. I said to her, you really should check the reverse potentials of these spots, because if you're close enough to the cell body, you'll get a clean reversal potential. But if you have a really, really bad cable problem, the reversal potential will get more and more negative artifactually so, because you have less access to that spot. So then she started routinely looking at reversal potentials. So here's one ganglion where here, far away from the cell body, she's a really nice spot, and she had a reversal potential somewhere around -80. Here, she had a smaller response and an apparent reversal at -83, -87. Here, there was no response, so there's no reversal. But what Adrian finally figured out, and I think, well, it's on the next slide, what she figured out was that in a given preparation, the reversal potential, almost wherever she checked it, was about the same, whether it was close or far from the cell body. And this is just one example, and I think this is not, I don't think these are the data for this cell, but this came by email last night. So these are distances from the soma that go from 150 to 700 microns. And the reversal potentials are within a relatively small number of millivolts, but most importantly, the more distant ones are more depolarized, if anything, than the next ones. And as she looks across the population, she sees a fair bit of variability in the actual numbers of the reversal potentials, but within a preparation, they're quite close, and this is the CV of those numbers. And so you can see within a preparation, all the reversal potentials are within a few millivolts of each other. And the slopes across 300 microns from all of them are plus and minus a little right around 0. So this is telling us that, in Adrian's words, that these neurons are remarkably electrotonically compact. And they're much more compact than we would have necessarily expected. The reason this is important is it tells us that it may not matter exactly where synapses are made. What may matter is that during development, the LP and the PD, which make a really strong synapse, they just have to find each other. But it may not matter which branch of LP and which branch of PD finds each other. So you may actually not care how you partition that neuropil space, but that you find your right partners. And it changes the way you think about the way this system works. And it's particularly interesting because most of the computation in this network works in analog. These cells release transmitters, a graded function of membrane potential, so these values are all consistent with that. Now, I'm going to very rapidly do homage to the experiments we did to start, or just a sample of the experiments we did to start looking at perturbation, to whether multiple animals could respond the same way to perturbation. And temperature is the best global perturbation we could think of, because temperature will change the properties of every single biological process, enzyme, receptor kinetics, ion channel kinetics. And people, biologists, not physicists, would calculate a Q10. I was trained to know that a Q10 was a Q10 of 2 would approximately say that a process would double for a tenfold, for a 10 degree change. A Q10 of 1 is a temperature invariant process, a Q10 of 2, and then there's an exponential here, so a Q10 of 3. Ion channels can have Q10s from 1.5 to 100 for a really temperature-sensitive process. And if you think about a rhythm, if you think about a pyloric rhythm where you have LP, PY, PD, LP, PY, PD, or me walking across the floor, you immediately realize that what matters is that you maintain the phase relationships even while the frequency is changing. And that turns out to be a non-trivial problem. It's a problem that you guys are working on here. So what happens when you change temperature? Well, Lamont-Tang measured crabs at 7, here's a pyloric rhythm at 7 degrees, beautiful pyloric rhythm, PD, LP, PY. And now that same animal at 19 degrees, beautiful rhythm, it's faster. We all expected that. And that has an apparent Q10 of 2.3. It's sort of a funky measure. But what we didn't expect is that the phase relationships are temperature invariant. So that means that while the rhythm is going faster, the relative timing of everything within the rhythm is maintained. And that's just shown again in intracellular recordings, where Lamont used the computer to scale time so he could superimpose the intracellular recordings at 7, 11, and 19, and that's shown here. And you can see they're basically superimposable, which tells you that all the processes that control phase are somehow or other scaling in a way so they work together. Now, the trivial way this would happen was if they all had the same Q10. They don't. We measured their Q10s. And so what we eventually realized is that the pacemaker ensemble has a duty cycle which is very well scaled, and then the follower cells have other mechanisms that allow them to do this. And I don't have enough time to tell you that whole story, but I just want to show you 2 more slides about this to go back to the compensation. So Lamont did an experiment which was never labeled, and I have another version of this slide which is not in the wrong talk, but you have the 3 pyloric neurons at 23 degrees, beautiful pyloric rhythm. He took it up to 31 degrees, the rhythm crashed, there's no rhythm. And then when he went back down to 23 degrees, the rhythm came back. And that's telling you that this was a crash, but not a cook. The preparation is fine. And so we realized that if you had a different set of underlying parameters, different mechanisms across animals, you might expect them to crash differently. And so that's what Lamont did next, is he took 5 animals, and he's plotted spectrograms here, where he's plotting from the power from 0 to 6 hertz, and each of the neurons, here's PY, LP, PD. The red means they're all going at constant period and the same period. So we've got animal 1, animal 2, animal 3, animal 4, animal 5. And at 15 degrees, they're all regular. At 19 degrees, they're still all regular, but they're going all faster. At 23 degrees, this one has started to show interruptions and crashes, whereas these guys look like they're pretty good. At 27 degrees, this one now has become very intermittent and has already crashed. This one is showing some crashing. These guys look like they're still pretty good. At 31 degrees, this one had completely bit the dust. Yeah, I think he turned off the recordings because it was so bad at 27. Graduate students do these things, right? Okay, but these other 4 that remain, you can see they're all crashing intermittently, but they're all crashing intermittently with different dynamics. In this one, LP more or less completely goes silent. In this one, you've got these bouts of good activity and crash. Here you've got bouts, but much different frequency bouts, etc. And as we look at each one of them, you can see they're not producing a canonical rhythm, they're producing a very different rhythm. And this is exactly what you'd predict if you had the effect of, you take something out of its operating range and push it to the place where those different underlying parameters are showing up. And I'll just show you the conclusions from that, from these data. At low temperatures, all networks are well behaved, and this is really important. The temperatures that the crabs would normally see, they all do fine with. They show consistent changes in frequency and outstanding temperature compensation. Each individual animal crashes with individual dynamics. And then I didn't show you these data. Some modulators expand the stable range, while others make the preparations more temperature sensitive. Now I'm going to indulge myself for about 5 minutes and show you one last point. Am I okay? For 5 minutes? Okay. He gave me permission. It's not my fault. Okay. Anyway, to go back to this question of homeostasis. So the components of functional circuits are not static, but are constantly turning over. So neurons living 100 years, receptors and ion channels turn over rapidly. And so neurons are constantly rebuilding themselves. And then the question is, how is function maintained? Now, when I first started worrying about this, and I was bugging Larry Abbott, we built our first homeostatic model. In this homeostatic model, Larry said, it's really quite simple. If you had some way of the cell could monitor its own activity, such as intracellular calcium, which we know tracks the cell's activity, and you use that as a feedback mechanism, so when the activity goes up, you would downregulate the inward currents, upregulate the outward currents. You could actually build a model that would allow a cell to tune its own intrinsic currents to come back to some stable point. And so we built a first generation model, which was published in Science and promptly forgotten and ignored. A second generation model, which had 3 sensors, which was published in Journal of Neuroscience and equally well ignored, but that's fine, you know. The Science paper is now cited much more than it was in 1993 when we published it. And be that a warning, right? Not to fall in love with Science papers. Anyway, but both of those papers were actually very useful for us in our thinking about the problem. Now, both of those models had pluses and they had minuses, and I'm not going to have time to show them, but they were both homeostatic in the sense both of them recovered from a lot of perturbations extremely well. They were very robust under certain classes of perturbations. More recently, Tim O'Leary, who was a postdoc in my lab, who just started at the University of Cambridge, built a new class of homeostatic models. And this one is much more biologically reasonable than the original ones. And what's really important about this model, and the reason I'm showing it to you, because some of you care about such things, is that the rates of channel expression are set independently for each channel. And I'm just going to very quickly show you what this model does. So here we have calcium controlling for each channel type independently the rates of synthesis, insertion, degradation of each channel type. So we've lumped that all into one rate constant. And that's just the equations that say that a calcium target gives you a change in conductance density, but now channel by channel. So if you start out with random low conductance densities and you run this model, the channels increase in density, and the cell here builds itself into a bursting neuron, and these are the channels that are in the membrane. It's a sodium channel, 2 calcium channels again, 3 potassium channels, and an IH. Okay, so random small numbers you build into a burster because the target was consistent with that being a burster. Now, the next thing Tim did is he did this over and over and over again with small numbers, starting with random small numbers. Here's one of those neurons building itself into a burster. But this is 6 different runs. Cell 1, cell 2, cell 3, cell 4, cell 5, cell 6. They all look the same, but this is on a log scale, so you don't see. They have very different, they have that range of conductance densities that we were measuring in our biological measurements. The conductance densities, each one of these has a different solution to being a burster, although they have very similar outputs. So this model is generating families of solutions, not individual solutions. And then the question is, what happens if you change the relative expression rates of the ion channel types? Okay, so now, here's one set of rates, and you end up with a regular spiking neuron with an FI curve that looks like this. Classic type 1. Here's a different set of rates, allows you to come up with what would be classic called a type 2 spiking neuron. A different set of rates give you a rebound burster. A different one, something that looks like a ventricular action potential. A different set of rates gives you just a tonically firing neuron. But, and here's why I had to show you that other thing. When you look in these models, now in purple we have now I'm plotting one of the calcium currents, the conductance, against the sodium conductance for 1, 2, 3, these different colors. You can see you have these strong correlations between the expression of these currents, and you also have that 2 to 4-fold range of values. So we're recovering from this model these strong two-dimensional correlations between conductance density that were present in our biological data that we never understood. And that's true for all these other ones as well. So we recover the multiple solutions, the spread in density, and we recover these correlations. So, and then I'm just going to very quickly, in one minute, show you what happens now if you do knockouts in this model. This model responds to perturbations homeostatically. It's quite robust to many perturbations. What is paradoxical is what happens in response to knockouts. So I'm just going to show you some of those. So here's cell 1, and we delete one of the calcium channels. And here the target calcium concentration is shown as a dotted line. The average calcium concentration is shown in the heavy green line. So we delete one of them, the cell goes off its target, and then the model runs and it comes back and it recovers its function. We take another neuron, which is from that same class. It's pretty similar in starting behavior, but now you do the same knockout and the cell goes silent. It's really sent for a womp. But now the homeostasis comes back and the cell recovers its function pretty well. So variable effects on two neurons that look the same from the same genetic pool, if you will. Okay, now here we have a wild type again and a knockout of the H current, and then the compensation, the model runs. It does it tries hard to get back to its target, but it can't make it all the way back. And that's because H deletions are really hard to recover from. But now we have a neuron, I think it's the same neuron, which we delete one of the calcium currents, it goes silent, and now it recovers back to its target activity pattern, but it recovers not to its starting condition, but this is an aberrant recovery. This is what you don't want. This is neuropathic pain, if you will. So it's come back to its target, but it's an aberrant recovery. And then I think there's just two more examples. Here's a neuron. We delete one of the calcium currents. It doesn't do very much. It compensates. You don't see much change. But we take that same neuron, delete the other calcium current. You let it run. There's not much phenotype when you do the deletion, but over time, the homeostatic process is running, and now you've destroyed the phenotype of the cell. So in this case, the functional homeostatic process can actually destroy the intrinsic excitability in response to this knockout. So just to end this, I'd just say that correlations arise automatically in self-tuning homeostatic models and depend on the rates of the tuning process. These models generate variability in multiple cell types. The homeostatic process may be working correctly and yet produce pathology in response to some perturbations and deletions. And this is really important because disease is often attributed to a failure of homeostasis. And this is saying that disease may result even when the homeostatic mechanisms are working correctly in response to certain specific perturbations with some sets of starting conditions. And the other thing I'm not going to talk to you about, these insights can be used to find neurons with the desired sets of properties. And I'd just like to end by just reminding you that multiple good enough solutions to circuit performance in the population are necessary to ensure a substrate for evolution under diverse environmental conditions. And I'm sorry I went over. Thanks.